right, as a reminder, if you're having difficulty with some of the material and you're not getting the material in class, if you will, you're certainly able to view, right, re-listen to the presentation, or obviously you could contact me if you have specific questions on content. But if you're looking for another way to study, don't forget you have that resource available to you in your Connect website in the lecture area. Just a review for you where we left off on Thursday, we were talking about energetics. We differentiated potential versus kinetic energy. We talked about the first and second laws of thermodynamics. We talked about reactions, whether they're endergonic or exergonic. And we contrasted true chemical equilibrium versus what I kind of consider like a set point or dynamic steady state within a living organism. And then we concluded with talking about oxidation reduction reactions. If you remember, it's kind of confusing, so I just want to just briefly summarize what it means if something is being oxidized. What does it mean if a molecule has been oxidized? Releasing electrons. Good. Okay. So if a molecule is being oxidized, it's releasing electrons. Very good. As well as it's releasing what? Energy. Okay. We could really just wash it down to that. When the electrons go to something, the other something's getting energy. That doesn't sound real technical, so technically if something is gaining electrons, it is being reduced and gaining an electron and gaining energy. So we're going to use these concepts in all of Chapter 5 and all of Chapter 6. We're going to see oxidation reduction reactions going on, right? We're just going to transfer energy from one molecule to another, in essence. So we've got to have an idea about that concept. Any questions about the difference between oxidation versus reduction? It's dealing with the transfer of electrons. You can't have one without the other. Okay, they're related. So therefore, you can call them redox reactions. In addition to transferring electrons, right, for transferring energy, a more efficient way to transfer energy is by utilizing this molecule called ATP. What does that stand for, ATP? Adenosine, actually it's close, adenosine triphosphate, but it's the same concept. It's a nucleotide, very similar to what we kind of crash course in our macromolecules right before the first test. So what I want to do is go back and review that ATP structure because we're going to be using it also throughout the rest of this semester. So I've got a little simple. All right. Which way is better for you guys to see this? Ball and stick model, the icon, or the space fill? Do y'all have a preference on? It's all going to be the same thing. Okay, yeah. The icon is real simple, obviously not structurally accurate at all, but you can kind of get an idea about it. Okay? And so if you remember the structure of ATP, we said it's going to have adenosine, which is our what? What's characteristic about adenosine? It's one of our bases that has nitrogen in it. So sometimes you call it like a nitrogenous base. When we get into DNA, we'll use that same type of prefix. Also, in addition to adenosine, there is what? Ribose. What is ribose? It's a sugar. That's very good. How many carbon atoms? Do we address that? How many carbon atoms does ribose have? 5. Okay, so it's a lot of times I'll abbreviate it just 5-C, or representing a 5-carbon molecule. And the really characteristic part about ATP is what? The 3 phosphates. Okay? 3 phosphates. And I'm fairly confident we talked about a phosphate. What is characteristic of a phosphate group? Phosphates, what's characteristic about them? It's found in the nucleotide structure. But what physical properties about the phosphate. You may have their notes from. Yes. It does release a lot of energy when the bond, not shown here, between the phosphates is broken, because what kind of energy, well, we know there's potential energy in there, but what about the phosphates makes it so reactive? Yes. Is another way to say reactive. Good. Did I talk about the charge at all of phosphate? Okay, well that's why. Just tell me no. All right. No, not at all. Phosphate has what type of charge? What's on your image in your handout if you've got it? What is the charge of each phosphate group? Negatively charged. Each phosphate is negatively charged. It is bound to a negatively charged phosphate and is bound to a third negatively charged phosphate. And you know that like products, like charged particles will do what with one another? They actually, if I have two positive, say magnets, and I try to push them together, will they attract? They actually repel, okay, oppose one another. So this is what's happening here. We have these negatively charged phosphate groups. They are held together by a bond, but do they kind of want, if I use the word want, do they want to be there? No, okay? And so did I use the word repulsed the other day, Thursday? They're electrostatically repulsed. You ever been repulsed by something? By someone's action? Repulsed? So there's an electrical repulsion. They want to get away from one another. Okay? That phosphate, that negative end on the phosphate is what makes it reactive. Okay? When it's bound to another phosphate. And so what happens is, once ATP is formed, which we'll actually see in chapter 6, it's not very stable because it wants to do what? Break apart. Okay? And it wants to transfer that phosphate out into something. And when that phosphate is transferred out, the thing that gets the phosphate gets energy. And so ATP is an important energy transferring molecule just because of the way it's made. Those are negatively charged phosphates. The cell's going to have to spend some energy to make that bond, but once that bond is available, it can be what? Once it's made, we can use it. Okay? We can transfer it from potential to kinetic energy. I want you to be familiar that ATP is not the only energetic molecule. It's not the only energy transferring molecule. It's just the most predominantly used energy transferring molecule. What questions do you have about this basic structure of ATP? 3 components include what? Do what? Adenosine, okay, and how many phosphates? 3, and a ribose sugar. They're all bound together. That makes up a single molecule.
What is the carbon of the ribose sugar?
There are 5 carbons, and basically each angle here represents where a carbon would be. So 1, 2, 3, 4, 5. Additional questions? We'll actually label sugars when we get into DNA, so you'll get really good at knowing where the carbon atoms are and how to put them in sequence. So that's coming up. I don't want to go there quite yet, though. Once ATP is made, we said it can be used. And what does it transfer out? It transfers out it's what? Phosphate. If I have something that's adenosine triphosphate, and then I get rid of one of the phosphates, the rest of the molecule is called diphosphate, adenosine diphosphate. And so ATP can be made or broken down to ADP, just back and forth. It cycles between the 2 molecules, 2 states, if you will. this animation should show you the relationship between adenosine triphosphate. Okay. This is looking at, I don't know what enzyme this is, but we can see our ribose sugar, adenosine base, and then we can have 1, 2, 3 phosphate groups on there. In this case, we're going to transfer the phosphate in the little reactive bond. Notice it's the phosphate at the end that's going to be cleaved. It's the one that's most repulsed. And so it's highlighted in this case in the orange. Energy is released is shown as a little yellow flash in this image. Oh, okay, that was super fast. All right, I'm going to back this up. So notice how this molecule entered in as ATP. And then where we're going is an enzyme can regulate these different types of reactions. And notice how ATP has become dephosphorylated. What does that mean, dephosphorylated? Given away a phosphate. It's lost of a phosphate. So now it's ADP. And now, did the other phosphate go away? No. It's actually going to get transferred into the other molecule. The other molecule has been what? If ATP has been dephosphorylated, the other molecule must have been phosphorylated. Okay, it gains a phosphate group. So ADP just got phosphorylated. What type of reaction was that? We formed a bond, released water, when we removed water, we formed the do what? Dehydration, condensation reaction, chapter 2. We formed, as a result of that little bond formed between the terminal phosphates. If ATP loses its phosphate, it releases energy, and if it's phosphorylated, it's storing energy, it's gaining energy. So it's just a molecule that can fairly easily add the phosphate or remove it. Okay, it's just a way to transfer energy. We're introducing the structure now. We'll understand how it's formed and used in Chapter 5 and Chapter 6 and the rest of the semester. So, yeah. Before I move on, though, what questions do you have about the structure of ATP? It's used for what? Energy transfer. The mitochondria can produce it. Also, the chloroplast can produce it, and it can also occur in the cell cytosol. But the bulk, you're correct. The majority of ATP in a eukaryotic cell is made in the mitochondria. So. What else? All right, so if we go back to our Chapter 4 handout, we talked about the basic structure of adenosine with the ribose sugar and the 3 terminal phosphates. We also identified that the phosphate can be removed, or it could be, the molecule could be dephosphorylated or added, and we call it phosphorylated. It'll cycle through between these two states. Where we're going, as I mentioned, in Chapter 6 and identified the mitochondria discussion is nutrients that you take in will should eventually be converted into ATP to do some work like we identified Thursday, mechanical work, chemical work, right, transport work, whatever, right, doing work within the living system. I may or may not have had you I don't think I talked about it, but it's on page 4. I don't want to get ahead of myself, but while I'm here on this image and thinking about it, ATP cannot be stored. And so I always should do what? We should always transfer it, we should always make it, we should always make it, we should always use it. use it or lose it. And generally speaking, a cell won't make ATP unless it needs it. Otherwise, your cell should maintain conservation, because you never know when your body might need more energy. You never know when you might need to run, whatever, right? Just think of all the scenarios that you might get into. Do what? So the terms that we described earlier is ATP, can transfer energy. If something is phosphorylated, what does that mean? If something is phosphorylated, gaining a phosphate. If something is phosphorylated, it's gaining a phosphate group, as well as gaining energy. So something's being phosphorylated, it's gaining a phosphate group, as well as it's gaining energy to do some form of work. Conversely, if something's phosphorylated, the molecule it's working with has just been dephosphorylated. Okay. Oh, sorry. Oh, forgot I had one of my awesome animations. You can see the terminal phosphates. I don't know if you can see the little bond I was trying to represent. I'm going to transfer the bond from the ATP over to molecule X. ATP has been dephosphorylated. Molecule X has just become what? Phosphorylated. Okay, so now it's just a transfer of this thing called a phosphate group. You okay on the terminology differences between phosphorylation and dephosphorylation? We also identified that ATP cannot be stored inside of ATP can't be stored. Okay, it's so reactive that as soon as it's within about a minute, if it's not used, it's going to spontaneously just cleave off at least that terminal phosphate. So it's not stable, which is good. It's reactive. It's good for transferring energy. However, that doesn't mean that cells don't have the ability to store energy. One way that cells can store energy is in the form of a molecule called starch. What is starch? It's a complex carbohydrate. It's a sugar. For example, what kind of species would do this? Right here? In the back? Did you say what kind of organisms, or did I hear the word? Did somebody say plants? What?
Yeah, plants.
Thank you. You have to talk that loud for me. I've got like old lady ears, so I need a hearing aid or something. So, yes, like plants. They can store energy in the form of this macromolecule called starch, which is a complex polysaccharide, okay? Or complex carbohydrate that you guys became familiar with. So sugars can be stored in plant tissue. Glycogen is the energy storage molecule in animal tissue. Again, a complex polysaccharide. What's another way that we could store energy? Fats, right? We kind of talked about those the other day. Fats, other forms of lipids, certainly are the ability to store molecules, store bonds, store potential energy. Very stable, right? But yes, it hangs around for years. We can store it. Those are some examples, certainly not the only way that energy could be stored. We can store it in the form of proteins and other macromolecules, but not in the form of what? ATP. Good. So ATP is an energy transferring molecule. We'll come back to it in chapters 5 and 6. Also, to understand energetic transfers in a living system, we need to understand the role of enzymes. Does anybody know what an enzyme, how we might define an enzyme? How might you define an enzyme? Very good. They're molecules that speed up chemical reactions. chemical reactions. The concise term would be catalyze chemical reactions. They generally are proteins, and from this class, we'll say they're proteins. And there's an exception, but the rest of our enzymes are actually proteins that work within or out in, say, in your body, like in the blood or something like that. They're molecules that carry forward chemical reactions. What's useful about enzymes is they are reusable. They're not just use it once and it's useless. Okay, I kind of joke about the terminology. They're green, right? Reduce, reuse, recycle. They're reusable. Once they're built, they can function for an extended period of time within a cell. In addition, our enzymes carry forward almost all chemical reactions that you will find inside of a living system. Almost anything that a cell is going to do metabolically, probably going to use a specific enzyme. So, for example, we go back to picking on our sugary drinks that we were talking about Thursday. If I want to catabolize the energy out of those, I'm going to use what? Enzymes. Okay. If there's sugars, I'll have enzymes that digest sugars. If there's proteins, I have enzymes that digest proteins, etc., etc., etc. Okay, so basically all our chemical reactions. What's interesting is it catalyzes chemical reactions that would happen anyway, but what? If these reactions would happen anyway, why do we need to spend energy in making enzymes? They're too slow. Enzymes are necessary for not only just carrying forward reactions. Okay, they could happen, but they would take too slow before the cell would do what? If we didn't have it, enzyme cells would die waiting around for something to happen. Okay? So enzymes catalyze chemical reactions that would occur anyway. An example is, right, does anybody have milk in their refrigerator? Okay. Like it? If I were to take the milk from your refrigerator and pour it in a glass and let it sit in the glass for a period of time, how long until those sugars just spontaneously degrade? Well, immediately just like, what is not long to you? Okay, so it might take a day or two or even longer, to be honest, to break down the sugars. Do you think your cells, if they need energy to be working out, should wait a day or two? No. Okay, so it doesn't make sense. And so where the analogy where I was going is there's sugar, there's nutrients in, say, for example, foodstuff. Those reactions would spontaneously, because of our entropy, they would spontaneously break down, but it would just take too long for the cell. It would just run out of energy. It wouldn't be able to do its work, and it wouldn't be able to survive. And so what happens is your cells are programmed to build specialized enzymes to carry forward specific reactions, speed them up so the cells can be responsive and survive, do all the things that it needs to do. That's just a general summary about enzymes. If we look at them in a little more detail, the way that they work is they express a region called the active site. And that name is pretty descriptive. This must be what? Where it's active, okay? Where it's going to do what? It's going to do the work. It's going to do the chemical reaction. Whether it's a catabolic or an anabolic reaction, that's where the combining of the reactants work to produce the what? If we look at a chemical reaction, our reactants are converted into our product. So the active site is the location of the enzyme where the reactants are converted into the products. Yes. The active site is the part of the enzyme where it binds, I have the words here, substrate, or I could put what? What other word have I just? Reactant. So it binds the starting material, binds the reactants, binds the substrates, and releases the products. This is important to understand because our enzymes are proteins, and we didn't get the formula. I didn't get to make sure you understand like complete protein structure. I gave you the presentation, you had the book, but I didn't really say the tertiary structure of a protein is blah blah blah. And does anybody know what the tertiary structure? What is the tertiary structure of a protein? And actually, it may be. But at least the tertiary level is probably the amino acids folded into the working unit. It may go into a quaternary level if there's multiple of these joined together. Generally, our enzymes are just tertiary level. I'm sure there's some that are quaternary, but work as just a single blob, if you will, of protein. So where I'm going is the protein, the bulk of it is maintaining this shape so that we can build the active site. Okay? The active site couldn't be built without the rest of the enzyme. Okay? So the rest of the enzyme gives the structure, holds it together. The active site is where the catalytic portion is of the enzyme. Okay? That's where, if you remember in the previous animation that I showed at the beginning of class, where ATP entered into that big green blob. The big green blob was an enzyme, and the molecule that it was phosphorylating would have and that thing would have been the substrates, and the output would have been ADP, and I don't even know what molecule that was, but whatever was being phosphorylated. Okay. So in that image, the big green thing was the large enzyme, but it was the animation was focusing on its what? Or zooming in on that enzyme's what? Active site. Here's an example of an enzyme called lysozyme. The protein enzyme itself is a light-colored purple structure, and this is an example of a tertiary level. It just means a functional protein. Okay? The little purple spheres represent individual amino acids. If we were to zoom in, we would see the little peptide bonds binding each of those little amino acids together. We know that when proteins are built, they fold. In this case, it folds into a glob. Does that word glob make sense? And a round, globby something. Some proteins form fibers, but our enzymes don't do that. Our enzymes are globs. But if you notice that the structure of the lysozyme, it looks like there's a little groove, okay, in there. What is that? The active site, right? So I want you to, if you've got your handout, I want you to label the enzyme, the active site, and the substrate. If you don't, you can just draw a really simple looking enzyme block. In this image, the far left is before and the far right is the after. Right? So I guess during really. That's that. It's the same enzyme, same function. Is the substrate the purple? Or is the substrate blue? What is the substrate? The substrate is actually which one? The substrate is the red item. Because notice how the red item can actually fit nicely into the pocket. Okay? And so the enzymes are usually really big, so they've got to have that structure to have that active site. And so what is the purple thing then? The enzyme itself. Only this little pocket pocket where what binds? The what? The substrate. Only where the substrate bind is the enzyme's active site. For some reason. Alright, I can't color on here for some reason, but if you can follow my arrow, can y'all see? That is the active site, and it looks like just a little, nice little pocket, for the substrate. On the image to the left, the enzyme has not bound the substrate, but to the far right, you can see how the substrate fits nicely into that enzyme's active site. What it's doing depends on the enzyme, the reaction, the metabolic pathway, whatever. Does that help?
Yeah, I guess I'm just trying to understand what happens when they bond like that.
Okay. So what happens when they bond like that is now we're probably going to stress a chemical bond or form a chemical bond. Your catalytic, your catabolic or your anabolic type reaction will happen. Okay, so it's either going to form a bond, break a bond, whatever the case may be. Whatever it's built to do. Does that make sense? Okay. So it's very variable. There are tens of thousands of enzymes. And so instead of giving you tens of thousands of lists of what would happen, you could just say it's either going to catalyze, or do a catabolic reaction where you're breaking, or an anabolic reaction where you're synthesizing something. Good question. Additional questions? So the substrate is colored which color? The red. The purple thing is the enzyme, and the thing that looks like it receives something is the active site. In addition, some enzymes need cofactors, and that name is kind of descriptive. Cofactors, these are factors that help out, actually necessary for the enzyme to do its work. Cofactors are molecules that a particular enzyme might need to do its work. Usually, but not always, certainly not always, let me see, these will, moving the wrong direction, have like a vitamin precursor. You always hear, got to eat your vitamins, got to get a good, well-balanced diet, because they help serve, some of them help serve in the role of coenzymes for your enzymes in your body to maintain your life, right, or the function of your different types of enzymes. Two important coenzymes that we are going to study or use include NAD and another one called FAD. If you on your list, there's a very similar molecule called, as I have shown here, NADP. I won't panic you and I won't tell you the name yet, but in chapter 5 we'll get you introduced to those terms. But you can put NAD, NADP, another one called FAD. Right now the acronyms are okay. But they get their precursor from vitamins or maybe other ions, right? Something like that. Metals that can facilitate or serve as cofactors for specific enzymes. So without them, the enzyme won't be able to do its work. Okay? So the question is then, what exactly is it about an enzyme that allows for it to do its work? We have an idea, it's got an active site, it binds to something, it carries forward a reaction that would occur anyway. And so the question is, how does it do that? Okay? And your textbooks always like to have these simple, good visual analogies. Okay? Simple analogies, good. If we look at a chemical reaction, and then take into consideration the amount of energy necessary to transform a reactant into a product, okay, whether you're doing a catabolic or anabolic reaction. Okay? If you're trying to get from substrate to final carry forward a chemical reaction. In this image, it's trying to illustrate how lactose, what is that? Sugar in the milk, okay? Lactose is an example of sugar in milk, and it's a disaccharide, so that's how many? Two sugars. Okay. And in order to extract the energy unit, that bond, that glycosidic bond, needs to be broken. As we identified, it could happen spontaneously, but it's going to take a long time. So the cell should have enzymes to help catalyze that. Okay? The amount of uphill energy necessary to break that bond is represented by this hill. Okay, someone trying to push a ball up the hill, representing the amount of energy necessary for that bond to just break and liberate this energy. You okay with the visual scenario? Just gonna see this little guy try to push a ball uphill. You can see he's really working. The steepness of the hill is pretty big. And so I was trying to illustrate how strong that bond is between those monosaccharides. What an enzyme does, it still does the same thing. It'll still take the substrate, still do something with the bond, and still produce the product. It doesn't change the inputs or the outputs. It doesn't change the fact that energy is necessary. What do you think it probably does? It makes it easier. Okay, so in this scenario, what would be easier? Well, so we're still trying to. In order for it to fall, though, you've got to. We could use that, but in gravity, it would pull it backwards, right? So what would make this scenario easier? If your job was to push a ball up a hill, what would you want about that hill? Not very steep. And that's basically what an enzyme does. Let me come back to this text in just a minute. What it does is lessen the steepness of that reaction. It doesn't change the fact that you're still working with the original substrate. It doesn't change the amount of energy necessary to change that bond, to break it or form it or whatever you're doing. In this case, we're breaking. It doesn't change the energetics of that bond. Okay? All it does is change the relationship. Okay, in this case. Notice how in the enzymes present, notice how it actually puts stress where the bond would be formed. There is, like physically separate. And it puts physical stress where the bond is, and then it causes that bond to break faster. Okay, in this case, we're looking at a catabolic reaction. Okay, we're releasing energy. There's still going to be energy necessary to overcome this state, the initial state. However, the amount of energy is less. So it makes the work more efficient as far as like energy and time and all the factors that you might want to take into consideration for it.
Impact of the reaction, basically.
I'm going to kind of hem-haw on my answer to that. In what fashion do you mean the impact?
Like, I guess I mean the work.
It lessens the overall work necessary to carry forward that reaction, yes. Correct. It's not wasteful, and it speeds it up. The proper term for that is called activation energy. Okay, so let's go back a page. Okay. What enzymes do is lower a particular reaction's activation energy. That amount of energy necessary to carry forward a particular reaction. So I make a note, right? Makes the hill shorter. Okay, if you want to go to your visual, right? Is that okay? Visual analogy makes the hill less steep. There are no hills in your cells, but do I? There are not. So it lowers a particular reaction's activation energy. You can shorthand write that as large E for energy and subscript A for activation. Activation energy. Lessens. Something else very, very, very important to this is that it allows for a chemical reaction to occur. It's controlled. It puts less effort. And this is critical right here. That energy is not transferred in the form of heat. Okay? So it'll carry forward a reaction without adding any thermal energy, because we don't want to have just reactions occurring spontaneously, releasing so much heat that the cell dies, overheats and dies. Okay? And so it controls the thermodynamics, if you will, to make them suitable for that living organism. Yes?
So that consumes
No, I wouldn't say that, and here's why. If you go back to the first law of thermodynamics, we said energy is never created nor destroyed, it's always transferred. And a lot of heat is always or a lot of energy is transferred in the form of heat. Okay, so heat, even if it doesn't have to be the magnitude that you can think of when you measure when you're hot. There is still heat when, say, we're below what we call freezing, like when we were negative whatever it was, 10 degrees, there's still heat in the environment. Chemical reactions are still occurring. It's just not hot to you. But so where I'm going is cautioning with your term, just perception of the word heat, I guess. Okay? And so it doesn't have to be to the extremes that you're thinking. Okay? And so, for example, let's go to the milk jug in the refrigerator. Okay? There's a ton of energy in it. I mean, the average person, it might take them, I'd say, at least 2 days to get through, at minimum. Okay? Or it might last you more. Okay? That's going to give you enough energy to store you through, or to get you through X number of days of work. Okay? If we were to break all the bonds and all the fats and all the proteins at the same time, so much heat would be released from that the milk would be hot. You wouldn't be able to touch it. Okay? And so all chemical reactions are going to transfer heat. What we want to do is keep it within normal levels. Okay? Safe levels. And so it doesn't add any extra heat to the system, causing problems for the cell. Additional questions or clarifications? All right, so we talked about enzymes lower the what of a reaction? Activation energy. Make the work easier. Figure 410 illustrates this for you. In the purple would be a particular chemical reaction shown, how much energy if it were to spontaneously occur. However, we can lower that activation energy by utilizing enzymes. You can throw any chemical reaction you want in here. You can talk about when your blood clots. Okay? If I want a blood clot to form, I need enzymes to start this step and that step or whatever. Okay? So normally blood clotting could happen spontaneously, but it's going to happen faster when we need it, when we use enzymes. Okay? So maybe we don't need to just hone in on like nutrition. It's easy to we eat, right? It's easy to focus on the nutrition, but any type of reaction, right? Making molecules, whatever, you just name it, enzymes are going to be used to produce products. Questions at this point? The term utilized to illustrate this, enzymes are called biological catalysts. Enzymes are called biological catalysts. They catalyze, they speed up chemical reactions by lowering that reaction's activation energy. So enzymes, good? Yes. Okay. Now what we can do is put them into perspective. Like, well, what you're saying, like, what are they used for? And they're used for different types of metabolic pathways. You've been on a pathway before, right? Like a physical path, right? What's a pathway? Like a trail, like a clear area, something like that. You have a starting point and then end point. Okay? But there's a journey, right? Molecules have pathways as what we saw with lactose catabolism, like have a disaccharide break a bond to products, the monosaccharides product. A lot of chemical reactions are much more complex, i.e., where we're going, chapter 5, photosynthesis, chapter 6, glycolysis, Krebs cycle, electron transport chain, blood clotting, you name it, they're complex, okay? And they're called metabolic pathways, okay? And all they are is just a series of chemical reactions that transform substrate, and it could be a single substrate, or it may be multiple substrates into a single product or multiple products. Just a series of chemical reactions. In order to get from point A to point B, each step is going to require a different what? A different enzyme. Each enzyme binds its own substrate and produces its own product, which would serve as a substrate for the next enzyme. Okay? And so there's lots of enzymes in many metabolic pathways. Okay? So each step is going to require a different enzyme. Some examples we've already listed is blood clot formation. Another example I think in your textbook is something growing like the leaf on a plant. Digestion, you name it. Probably a metabolic pathway involved in that process. Not all metabolic pathways have the same number of enzymes in each step. It just depends on the pathway. The way that these metabolic pathways work are by two primary ways. The first way is called negative feedback, and the second is called positive feedback. What do you think negative feedback, what does that word mean and conjure up? Kind of sounds bad, but this is a normal thing, so I don't want you to use the word, I don't want you to correlate badness with this. So what else was used? What other term was used? Go to electron. No. In negative feedback, the word that I want you to associate is opposite or opposed. In negative feedback, I want you to use the word opposed. What that means is, if we accumulate a product, the metabolic pathway has done its job. Should we keep the pathway going? No. Let's turn it off. Or the term is called negative feedback. So if we overaccumulate the product should do what to the first enzyme? It should cut it off. Stop it. Okay. What if my product is underaccumulating? What should the first enzyme do? Speed it up. Start it. So negative feedback isn't always just an off. It may be an on signal. But it's doing what? It's opposite. Okay? If something is underaccumulating, we need to carry forward that reaction to begin to bring that factor of product back to set point. I want you to clarify here. The product stops the pathway. I said it, but I want to make sure you write it down. The product stops the pathway so the product is in set point normal ranges or homeostatic ranges. So let's clarify that. Yes, the product stops the pathway, not to be bad, but to keep that product in homeostasis, in balance, in normal range. So negative feedback, I think, is okay to understand. If you overaccumulate something, we'll stop making it. If you're underaccumulating something, we'll start producing it. Let's carry it forward. However, some metabolic pathways rely upon positive feedback. What do you think happens during positive feedback? Not only just the same, it's instead of stopping, it increases, continues. In positive feedback pathways, a particular reaction continues. And so the product says, keep making me, keep doing this. So positive feedback continues some pathway. At face value, it sounds bad. You're like, well, you're making product, and you're making more product, and you're making more product. So at face value, it seems like you're going to get far away from what? Homeostasis. But sometimes we need to start something, and we need to keep it going. Textbook example, positive feedback mechanism. Anybody had a baby in here? Anybody? Or a couple of people? Uterine contractions. You want them to start and just keep going, get that baby out. Okay? Positive feedback mechanism. Okay? Not the only example, but a really good visual. Right? Positive, positive, positive. The blood clotting example. You want to start that clot formation and then keep it going, right? You've got an injury, right? You want to form that. You want to start that clot and keep it going. Right, those are some brief examples of positive feedback. Keywords. Negative feedback does what? Opposes, opposite, to keep something in homeostasis. Positive feedback actually keeps something going. Okay? And again, it's to bring something back to homeostasis. They're interrelated. I want to elaborate a little more on negative feedback, though. Okay. Oh, yeah. It's kind of like the balance, the homeostasis visual, right? If our product overaccumulates, we should convert it back to substrate, and then vice versa, right? Teeter-totter, homeostasis. Okay, so it opposes. Hopefully this image visually kind of helps you. The opposing. You can just do the reverse arrows, to be honest. Right one. Don't worry about drawing it. You don't. It's not great image, but okay. So negative feedback. Can you see the opposition implied in this image? Try to bring something into set point or homeostasis. Positive feedback. Let me come back since I'm going there. Let me come back. Positive feedback will actually keep something going. Okay, so the substrate continues to make product. Let me go back because I kind of got off to my thought path. Let's look at negative feedback in better detail. In negative feedback, as we identified, there may be multiple enzymes in a particular pathway. They're not probably going to be lined up all cute like this and neighbors. Okay, but this is easy to see them together. Enzyme 1, 2, 3, 4, right? You could have 10 enzymes or more that you're going to put in your pathway. What we said is you're getting from point A to point B, and there's stops along the way. The initial substrate, we said, will form a product which serves as the what for enzyme 2? Substrate, which produces a new product, which is now the substrate. Okay. Some terminology that you want to add in your pathways, on your metabolic pathway sections is the products in the middle, you can call those intermediate products. What does that mean? Intermediate. I-N-T-E-R in the middle. And so the products in the middle. In a metabolic pathway, intermediate products that serve as substrate for the subsequent enzyme. But we're trying to get down to the what? Not the intermediate products, but the final product. So, final product. Might want to differentiate the final product versus the intermediate product. So which color is the final product in this image? The brown. Starting substrate is red. Intermediate products are colored pink and white. Okay, variation. I'm just running you guys all out today. Is there like a party going on out there?
Did we pass around the information?
Thought so. Nope. I'll get it around in just a minute. Intermediate versus final. You okay? The way that this works, the way that negative feedback works, is by what's called competitive or non-competitive inhibition. I think you can handle the word inhibition. What does that mean? To inhibit, to oppose, right? And what happens is, you ever been competitive? Or not competitive? Molecules are described as being competitive or not competitive. What this is in reference to is an enzyme's active site. If a product is considered to be competitive, what that means is the final product binds to the first enzyme's active site. It competes with what? The final product binds to the first enzyme's active site. It competes with that enzyme's substrate. In competitive inhibition, not only does it bind the active site, but it competes with that enzyme's normal substrate. By converse, right, non-competitive inhibition, the product must bind where? At the end, the first enzyme, where? In non-competitive, it would bind somewhere else that's not the active site. So in non-competitive inhibition, a product can bind on one of the enzymes in the pathway somewhere other than that enzyme's active site. So that when that product is bound to somewhere else, it actually changes the shape of the enzyme, and most notably, it actually changes the shape of the enzyme's what? Active site structure.
So, an enzyme can have several substrates.
Yes. Yes.
But now one has active site and another.
And in that case, you wouldn't call it a substrate, but you're on the right track. It has several binding sites, is probably where you're going. You wouldn't technically call them substrates because they don't bind to the active site. They're usually called like inhibitors or I forget their word is, but they're activators. You can either promote or inhibit the active site. But yes, certainly enzymes can bind to multiple molecules. Okay. Your figure 413 in your textbook illustrates an enzyme binding to its substrate to form its normal product. In competitive inhibition, go to the far right image, in competitive inhibition, where it says inhibitor, that would be, say, the product of that pathway. It's labeled as inhibitor, but it would say the product. If the product binds to that enzyme's active site, can it bind substrate and make more product? No. So that enzyme is shut down for that moment, and the pathway is turned off, or we just said negative feedback is happening. So the key thing is competitive, a molecule binds to an enzyme's active site. Non-competitive inhibition is where binds somewhere else, and that somewhere else ends up changing the shape of the active site. So even though it's not competing with it, it's changing the activity of the enzyme, so now it can't do its job, so we still see negative feedback taking place. Those are some ways that enzymes can be regulated. And we also contrasted that to positive feedback. Some examples I've already given, like how this enzyme called lactase, which is illustrated by this colorful ribbon structure in the background. I was looking at the fine structure of this enzyme. Lactase will take lactose, the disaccharide, and catalyze it so that that glycosidic bond is actually broken. And the products would be two monosaccharides, galactose and glucose. These, for example, could be further catabolized using other specific enzymes. And we kind of went through a pathway already how the substrate forms a product, or this converted product can serve as a substrate for another enzyme. The remainder of Chapter 5 is talking about transport, and that's what I had you guys complete on your worksheet. And we really don't have time to discuss it today. And so what I'd rather do is spend the last 10 minutes going over your exam. Okay? So why don't you go ahead and put your materials away. I'll stop recording here.